You know, we were looking at family pictures. My sister got a whole bunch of family.
Very impressive.
And it shows people that were around 65.
Not that I know of. No.
They were old. They were old. And their hairstyle was old. So they may have lived to 95, but they looked
But, you know, we moved them to a retirement complex in Binghamton, nearby the system. And I had already planned to go visit him. We would sell his country place. So I was already planning to do that. So I'm still going, but I'm going to help the sister. Yep, it's getting our generation now.
You come across the term thyroid in your thyroid. MicroRNAs are basically I've never seen the word I didn't talk. Thyroid, I've never seen the word virus. Yes, and that's how they don't affect humans. They never ever go.
That's really scary. Because that's, you know, when I had my
Some down in Canada, but they just become, you know, it's kind of like smoking.
when he was on dust.
And work our way down.
Oh, no, this is great. No, it's exciting stuff. And you're right, I don't know.
Well, actually, that's not
And very repairable.
Yeah, but that's what I meant.
Oh, Washington every
That's today.
Oh,
But they don't have
Yeah, and
Tesla or any of these.
Hey, they're having an open house Saturday.
I could drive your car. Just went with you. I could get a free test drive.
I know that we want to get.
Would you let me drive it? Oh, sure.
So long as you don't start the battery on fire.
Yeah, I have to run over a big piece of junk and start the battery on fire, then shove it up into the
We got to
frame.
get everything done, and then he's not going to.
We were in D.C. last week.
You're hired. And he does a lot of electrical work.
Second gate.
Pollen counts in Alaska. And what do they say?
It's like 3,000.
I was gonna say it's way higher than here, but it's only for like 6 weeks. Trees have to be close to here, but we've cut down enough trees that and grass, I don't know.
Democrats are going to lose a lot of votes.
You don't hear one like that.
Give it a few more minutes there. Their policies are going to come back.
So a reverse snowbird.
How did you pick this topic, Ms. Lammert? How did you pick this topic?
I knew she would handle it well. And I knew I knew nothing about it. I didn't understand it. Someone had to
Have to
translate
do to you what it's done to me over the years as I states and weird.
Not as creative.
I don't know about that.
Holy Grail or a puzzle
sending me all
piece.
these emails. They have to delete it. I get like 50 emails from
Primitive block.
you have to take it one for one each.
Yeah, you go in, well, you can do a whole bunch of work. You go in, and you have an option of blocking all future emails from that person.
I'm lazy, I just delete them.
He wants to know that he wanted to like to have it.
It's like when you get real mail, you get excited for that mail to be in real hall.
Grocery weekly.
It's like when Kramer on Seinfeld decided to take all the catalogs out.
I didn't see that one.
Supposed to have 4 fellows, so we'll see if a few more show up.
And I will get it.
Yeah, she either just leave it at you could leave it at send off. Yeah.
Thanks for doing that. Sorry.
So there's a bunch of things that there's
What do you got blooming? Siglala? Do you have a lot of things?
okay. Hydrate.
So Mike, when are they going to come out with four-wheel drive this year?
Just go on their website. It has the timeline.
When is it?
This year.
Oh,
I love gardening.
then I'll be interested.
I'm having an open house over here this weekend. But they won't have to.
Yes, I've decided I don't ever want any car other than all-wheel drive. It's just a pain.
I was in DC last week. All the Israelis were out.
If I get another Super, that'll be my 10th.
We missed very long.
I have to be prepared to go into double digits.
Yeah.
But my kids wrecked 3 of them, so it's not.
Yeah. Yeah.
Aaron's on vacation.
Oh, yeah. Frederick's Vernon? Yeah.
Now that Frank is here, we can get started. That's right.
David?
Part of the U and
The word of the day for those claiming CME is exosome. Zoom, that's in your talk. The only other comment is we have one more session here, and then the last session of the year is on the 29th, and Jerry tells me so far the number of RSVPs is fairly low. I don't remember if I sent out, you remember if I sent out an announcement of our last session? I did? I'll send it out again. I didn't think I did. Maybe. Anyway, that's an evening session on the 29th over at Polaris, what I used to call, or it used to be Battelle. So it should be a very enjoyable social evening with not a whole lot of heavy scientific stuff. So I will send out an announcement today with all the specifics, and go ahead and RSVP as soon as you can so you have some idea of the head count for that evening. So today, Joyce is very appreciative that I gave her this topic of MicroRNA in allergy and immunology.
The solution to everything.
That works.
It's this little-known story, little-known fact that Paul McBride and I actually practiced together for a couple of years. And when I started out, he assured me that, they just give the old guys clinical topics. And I've always felt safe about that. And then I got this topic and had to Google it to figure out, thank God for the internet. Anyway, about the same time I was watching Lost in Translation with Bill Murray, and that's what this reminded me of. I felt like an American who didn't understand the language. And it turns out, as you'll find out from this is really what this is all about, really stopping translation. No disclosures. MicroRNA actually was first discovered in 1993, and it identified probably around the early 2000s as kind of a key regulator of proliferation, differentiation, and cell death in both normal cells and in aberrant pathways. And when it first came out, it's like every one of these other topics that we get, is this the holy grail? Is this really going to be the key? Or is it just another piece of the puzzle? As you'll find out today, it's probably just another piece of the puzzle, but they're very, very intriguing, and I think there's lots of possibilities. And I know that there are researchers all over the country and all over the world that are just busily cranking out data around this area. So this won't be the last time you see this. I had to go way back. This is when I took Biochemistry 101 so many years ago, I can't even remember it. It was central dogma. You have your DNA, and it undergoes transcription in the nucleus, and you get your messenger RNA, goes in the cytoplasm. You've got your transfer RNAs, the ribosome, and you get protein. And it seems so nice and linear and simple when I learned it back then. And as everything that we talk about here, you learn that there's nothing that's simple and linear and easy to understand. And it turns out that really making proteins is really this very complex regulatory network, lots of things talking to other things. And so as I went back to remind myself about messenger RNA, there are a bunch of things that happen. The first thing is there's this 5' cap, which is modified binding nucleotides that are added to the front or the 5' end. And the presence of this cap is really critical for recognition of the ribosome and for protection, particularly from RNAs, because these messenger RNAs can get chewed up with RNAs. The cap is added just at the time that transcription starts happening, and so that the cap actually influences the transcription and vice versa. Splicing, another thing that didn't exist when I took Biochemistry 101. What you see in a messenger RNA isn't all protein. So you've got introns, which are non-coding areas that are in that messenger RNA. Then there are stretches that actually are important called exons that are the protein coding sequences. And then it turns out that sometimes what we call this pre-messenger RNA can be spliced different ways. So you can have shorter proteins, longer proteins, and that's called alternative splicing. It turns out that splicing is performed by an RNA protein complex, and that's called spliceosomes, and that could be the word of the day. I like that one. And then it turns out that messenger RNA is actually fairly creative and actually carries some of this capable of actually catalyzing its own splicing. So they carry their own enzymes there, which are ribozymes. So very complicated stuff. You add to that the fact that messenger RNA, that there's editing that happens. So depending on the cell that the messenger RNA is at, sometimes different nucleotides will be added in, and so you'll get early stops, shorter proteins, longer proteins by this process of editing. And then there's polyadenylation. These polyadenyl moieties are added to the end, and this happens to all except the histone messenger RNA. And again, very important for a bunch of different reasons: for stopping transcription, for keeping it from getting chewed up, and for export of the messenger RNA actually out of the nucleus. And then there's transport. It turns out that in plants, you don't have to transport because it's all one thing, but in eukaryotes, it's in the nucleus and then has to be transported out. And there's special proteins and signals on the messenger RNA that allow messenger RNA to be transported. And it turns out that the way that it happens is different at different cell types. So it's different in a brain cell as opposed to a heart cell. And finally, the translation. It occurs on ribosomes, and they can happen in the cytoplasm just free-floating or in the endoplasmic reticulum. So what seemed like really simple when I took biochemistry has actually got all these really cool little things that tweak the messages and the proteins that you get. And more recently, they've discovered this whole family of what we call small RNAs, and they probably play very important roles in a variety of different things, from differentiation to stress reaction, and they're certainly probably important in diseases. There's a bunch of different ones. The small interfering RNAs, which are double-stranded RNA molecules, 20 to 25 base pairs in length, and they interfere with the expression of specific genes with complementary nucleotide sequences. And they function by causing the messenger RNA to be broken down after transcription. And the thought on these small interfering RNAs is they probably evolved as an antiviral kind of function. Then there are the PV interacting RNAs, and those are the largest of this group, large being a size of 26 to 31 nucleotide pairs, so they're not very large, larger than the rest. A big family, They probably probably involved in a lot of silencing of genes, and they differ actually from the microRNAs in a couple of ways. And one is that they're not as evolutionarily conserved, and secondly, they have lots of other complexities in how they're processed and what they do. Unfortunately, I don't have to talk about those because the one we're going to be talking about today are the microRNAs. So this is Caenorhabditis elegans, and it was in 1993 where the microRNA was first discovered in this organism, which is a 1 millimeter roundworm. It's a non-parasitic, found in the dirt. It's transparent. It's been used probably since the early 70s in a lot of molecular biology. But one of the things that they discovered in 1993 was that there was a small piece of RNA that could totally disrupt the development of these worms. And that's sort of how it came about to be discovered. So it was sort of back in 1993, but the research that really started cranking around this didn't happen until around the early 2000s when they really started looking at these things. So what are they? Well, it looks like that got messed up a bit, but they're 19 to 25 nucleotides in length. They're non-coding RNAs, and their predominant role is probably to mediate post-transcription silencing of the target genes. And unlike the peewee RNAs, they're highly conserved in nature, both in plants and in eukaryotes. They recognize a distinct class of small regulatory RNAs, and as they've looked and done a lot of research, they seem to regulate lots of different functions. Cell proliferation, differentiation as in the roundworm, apoptosis, stress reactions, probably important in immune responses we'll talk about in a minute. So how are they made? Well, we talked about that nice, straightforward Biochemistry 101. These are a little bit more complicated. So they're located in introns, exons, or inner genes in the genome. And they're transcribed by DNA polymerase II as a large primary, what we call PRI, pre-microRNA. Then they're processed by this thing called endonuclease drosha, and it goes into a hairpin structure, and that's called precursor or pre-microRNA. It's then transported into the cytoplasm, where it's cleaved by endonuclease dicer into a single-stranded, mature microRNA. And then the way they work, they directly suppress gene expression by base pairing to the 3' untranslated region, or that UTR, of the target messenger RNA. And depending on the level of complementarity to the target cell, they can degrade the messenger RNA, they can stop the translation, or they can do both. It's kind of their mode of action. So this shows you kind of in diagram form. So here's your DNA, ribonuclease RNAs, 2 transcribes, drosha turns it into this little winged thing. There are export mechanisms, so it ends up in the cytoplasm here, and then this endonuclease dicer cuts off the little loop end, and then it forms proteins. You can't really see the proteins there, but this RISC stands for RNA-inducing silencer complex. So it binds to this messenger RNA, and then in this particular case, it's blocking translation. But it turns out that as they've learned about these things, there's lots of other cool things that they can do. They're floating around in this. Drew said this is tricky, and he wasn't kidding. They can get in these like micro vesicles here and be excreted in exosomes, which is your word for the day. They can bind to these proteins that we're going to talk about called Argonaut proteins, which are really cool guys, and be excreted out here. And this is important because this protects them from RNAs. And they can actually bind to proteins that are outside, particularly HDL. And again, they can be carried to different places, and the protein protection from RNAs. And they've looked at like half-lives of these guys, and they can vary from hours to even days, depending on the tissue, like liver or heart. And then there are very short half-lives, like in the retina, in the area where you adapt to light and dark. It may be only an hour that these things last. But I think the important things to get from this is the fact that these guys can float around in the bloodstream, and they can have really long half-lives. They can actually go to different places, different cells, different parts of the body, which is really cool. Oops. And this just shows you a little bit. So here it's coming out of the nucleus, and here's this little looped thing here. And then the enzyme clips off this end, and these are those proteins that it starts binding to here. This is in plants, and in plants there's perfect complementarianism, whatever, from these little small RNAs and the messenger RNA. And in eukaryotes, there's usually a little bit of a mismatch here as it binds. It has these proteins that are very important. So this is what would be called this RNA silencing complex that ends up stopping translation and degrading the RNA. And this just shows, it turns out that there's multiple ways that this, here's this complex, so this would be the small RNA with proteins attached to it, binds to this messenger RNA, and it can have a variety of different ways to mess things up. It can block initiation factors, which is M1 over there. It can block the small ribosome unit from binding over there. It can block the large ribosome, which that M2 mechanism. Various mechanisms where it stops translation, which would be M3, and then M7 and 8 there are ways that it works to degrade messenger RNAs. So lots of different mechanisms once that complex is formed that can mess up RNA and stop protein from being made. So, like anything in nature, microRNA has built-in backups. A single microRNA can target hundreds of genes. A single genes are targeted by multiple microRNAs. And then they can also exert global effects on gene expression because they can actually affect epigenetic mechanisms like DNA methylation or histone acetylation. So that can be a really fast way to affect things. They also target transcription factors. And then, as we talked about, because they're in these vesicles and they're bound to proteins in the plasma, they can actually go to other places and have impact as well. Unique features of microRNA versus messenger RNA, 11% are encoded by multiple genes. Multiple microRNAs can be produced from a single pre-microRNA. A single transcript may encode a cluster of distinct messenger microRNAs. See, the largest one there has more than 50 potential microRNAs located in this one domain on the DNA. As I've learned more about these, and there are people in labs busily cranking out these all the time, they've been categorized into families according to homology of their seed sequences. And as we said, one microRNA can target many different messenger RNAs and the converse. So different in the same compared to messenger RNAs.
Do they exist in organisms that don't have DNA, that just use RNA for replication?
I don't know. That's a good question. Let's read this. This is a mathematical modeling of microRNA-mediated mechanisms of translation expression. We're going to show you more of these. You can go online and you can actually download computer programs that allow you, if you've been doing research in this, to try to, because they collect all these microRNAs and then they try to go back and find out what are the gene targets, or where do they bind on messenger RNA. We've already talked about the fact that even on a regular piece of messenger RNA, you've got like all of these different genes that can be coded in the same area. And so they really have to use this mathematical modeling. And one of the things I really like about this is you start reading this stuff is like, when I studied biochemistry many years ago, everything was very linear, and then you started looking for these connections. And at this point, really think about networks in almost this three-dimensional way that the proteins and the genes and the messengers all interact. So science is so much more complex now and so much cooler that it's amazing. So where are they? Well, they're in cells, but as I said, one of the things that really intrigued me, as somebody who really is into biomarkers, is they're detectable in cell-free body fluids, so serum and plasma. They're protected from the blood RNAs by forming the complex, as I said, with HDL. They exist in the cell membrane-derived vesicles, or your exosomes, and they can form complexes with these Argonaute 2 proteins, which are part of that RNA-induced silencing complex that's responsible for gene silencing. So they can do a lot of different things. And they were also very, very hardy. I read that they're passed in breast milk, which is very acidic. And when they purify these things, they're pretty resistant to heat and cold when they purify them. So they're really hardy little guys. They hang around a lot, and they're small. So when I was reading the literature initially, it was very difficult. The techniques to isolate these are pretty complex, and you can get a lot of noise in the system when you do this because they're such little tiny things. But they do have a lot of techniques that make them pretty reproducible at this point. And so these are the proteins that bind there. They're called Argonaute proteins, as Argo was the ship that Jason had for the Golden Fleece, not in sailors. So these are, which I thought was really cool. So these are like a little Golden Fleece. And it turns out that the number of proteins that are in these complexes varies. In humans, there are 8 different proteins that can be found in these complexes. In yeast, there's 1 protein that's found associated with these microRNAs. So the families of proteins in humans, there's these ones called ago-clad proteins, which are the ones that you see most of the time in the diagrams. There's peewee-clad proteins, and then there's specific clad proteins called wagos. And there's huge amount of work that's been done to some of the proteins on the structure and the function of them. And I actually found several reviews just on the proteins themselves. And this just reminds you again, here you've got a little hairpin exported with exportin 5, binds to this ago protein, split by the little end taken off by the dicer. So you've got this little complex here. It goes from double-stranded DNA to a single-stranded DNA, and the other strand is chewed up, but they don't know really the mechanism for that. And then it goes here and does its job, and then alternatively it can go out into the plasma in protected forms and do other sorts of things. So the important roles for these guys, and why we're talking about it today, is they've been found in a variety of different things. Development. As you recall, the worm that we looked at, roundworm, was one of the first places where they found that the microRNAs actually disrupted the development of these worms. In animal models, they can actually, if you cut these things out, it can cause embryo lethality, so it's probably important in that. There's a huge amount of work that's been done with cancers, and lots of these are actually used as biomarkers in different types of cancers at this point. Autoimmune diseases, there have been places where dicer deleted in Tregs actually resulted in mice models of fatal autoimmune diseases. They're important in cardiovascular disease. And then finally, there's been a ton of work done in the last probably 14 or 15 years in allergic disease. And this just shows you in cardiac disease, when you have an MI or you have CHF with stress on the heart, there are actually these microRNAs that are released either from the death of cells or actually actively released. They go to other cells, but you can actually measure these. And again, it could be another marker, for example, CHF or for a cardiac disease that you can measure. And you can tell this is the American Heart Association with posters on microRNA. Its time has come. It's not just the next new thing. And this is there were 2 or 3 brand new reviews just from spring of 2014 on cancer. But this just shows you, this was from Nature in 2005, and probably does play a significant role. If you've got the normal cell where this microRNA is actually regulating the target, keep growth under control. But it can block cancers, or it can actually maybe bind to the oncogenes and start cancers. And there's a lot of work being done looking at these things as possible therapeutics, and again, as possible biomarkers for different sorts of cancer diseases. So allergic inflammatory disease is obviously like a great target. People were really excited because, we've got this Th2 type with predictable histologic findings, molecular changes in genes and proteins, and pathways that would be ideal for fine-tuning with these kinds of processes, and that's the reason people were excited about it to begin with. And this is sort of a summary slide. We're going to talk a little bit more about microRNA 21 and allergic asthma, but most of the studies that have looked at allergic asthma have really been in animal models. And the models using kind of that shared phenotype, airway eosinophilia, the Th2 inflammation, and airway hyperreactivity in these models. And when you look at that, this microRNA 21 is actually one of the top overexpressed microRNAs in inflamed tissue. Its highest expression is actually localized in macrophages and dendritic cells. And the thing that's very exciting is this IL-12p35 pre-prime untranslated region actually harbors a very highly evolutionary conserved target that's for the sequence of microRNA 21. So it's an area right where it binds. And it's kind of a key molecule involved in this Th1, Th2 polarization. And so the question is could that be one of those keys that actually moves you one way or the other? That's one of the things that's very exciting about this. And so these are these heat maps that they do. And make sure I've got the... so this was an animal model for IL-13. And in this particular animal model, if you give doxy, IL-13 gets expressed. And without doxy, it doesn't get expressed. And so in this particular one, they took these transgenic mice that express IL-13, and then they gave the doxy or not. And what they did is they took probes for mouse microRNAs. And at the time that they did this research, which I think was 2009, there were over 579 mouse microRNAs that were available through the microRNA mouse registry, which is pretty cool. And over 121 of those 579 were expressed above background. But when they did gave the doxy and the IL-13 was expressed, these are the microRNAs that lit up. You can, the greens are underexpressed. You can see up here. So those would be ones that were repressed, and the reds are the ones that are upregulated. And you can see actually, I think. Let's see.
What's doxy, Joyce?
Doxycycline.
Oh,
yeah. Yeah, this one right here, the red one here, so that's overexpressed, and that's microRNA 21. And you can see up here in the greens, the underexpressed microRNA R1 is underexpressed on that one.
When they number those, it just that relates to a structure, a fixed, unique structure.
Yeah, yeah. And when you think about, like, so you've got 500, talk about needles and haystacks, so you start it with just this massive stuff, you throw in 579. To me, this is pretty amazing. And so, this is kind of the relative overexpression, and this is without doxycycline and with doxycycline. Remember, with the doxycycline, you get the IL-13 expressed. And you can see that microRNA 21 is just like way overexpressed. There are other ones, for example, here, that are overexpressed, but this was really the predominant one in this model. And then this is the microRNA 1, which was significantly underexpressed.
What does doxycycline have to do with it? Why does that change?
I don't know. Reading the materials and methods in some of these were way over my head, and so why that induces IL-13 in these transgenic mice, I don't know. Maybe Bill
Well,
can
they
tell us.
have in the transcript, it's sensitive to doxycycline, so they'll turn off or on a gene and you express it.
They engineered it that way? Or?
Yeah.
Oh, so it's not naturally that way.
No, it's all engineered.
Yeah. These mice.
Doxycycline is engineered, or they?
No, they put in a response element that's sensitive to the doxycycline. And it'll splice out the gene or not, depending on which way they want to overexpress or underexpress it. It's sort of like when they put these fluorescent probes on if IL-4 is being secreted by a gene. In the original transcript, they're like sensitive to an antibiotic, so you can splice that into the actual germline, the fluorescent protein. It's a complicated thing right at the very beginning of the stem cell level. But it's a nifty way to monitor gene expressions without interfering with all the other regulatory enzymes and things like that.
Yeah.
But the doxycycline is a long time. It's just an artificial way of
It's just
amplifying
like a
your,
switch.
or shutting off a particular gene of interest.
When you start reading these papers, and in the olden days, I used to be able to read materials and methods and sort of understand, and you realize that this is the age of miracles and wonder. There's just things that are amazing. You can just actually order off the shelf now to do this sorts of research. Bill can tell us all about it. So stop me at any time when I make major blunders, okay? And they've looked in the bone marrow in this particular study, and you can see that it was overexpressed, particularly in bone marrow dendritic macrophages. And then the BAL cells, type 2 alveolar macrophages, is where they found this microRNA 21. So this was another by the same group, Thomas Liu, I think. And this was, I think, 2011, so different animal model, but still mouse. And this is ovalbumin sensitized mice that actually have the typical phenotype of eosinophils in the lungs and bronchial hyperactivity. And the same sort of study here. So, but here you have mice that are negative for microRNA 21 and mice that actually have microRNA 21, or a flip of that, and non-sensitized and sensitized, and then they do the same sort of study. And what they found in these negative mice is this kind of interferon gamma signature genes that were turned on in these microRNA negative mice that couldn't produce the microRNA. And they found out in these deficient mice that they actually did have reduced lung eosinophilia after allergen challenge. They had increased interferon gamma, and they had decreased levels of Th2 cytokines like IL-4 in the BAL fluid in these mice. So all the things that you would expect in the Th1 versus the Th2 in this particular model. And then this is they use this is called an ingenuity pathway analysis, and they took the things that were upregulated, which would be the interferon gamma and the IL-12, and they look at phenotypes and gene products, and look at the co-regulation of the interferon gamma and IL-12, and in this particular way. And like I said, it's like more really looking at this global pathway. And then this is kind of the next.
You can't get away with just one.
So this is sort of the next step of this, and this is called top cluster network analysis.
On that other one,
Yeah.
ingenuity pathway analysis, how they do that, they use a company that, hired a whole bunch of people to go through the literature and find out what were the protein targets of various, all the genes regulated in the literature by IL-12. So they put that into the computer.
It's a computer-generated thing telling you the outcome if you upregulate.
Yeah, and then they give you a probability estimate of what. So they've got they know what IL-13, all sorts of mucus genes and airway hyperresponses. So they keep going through the literature and kind of making, just like Google, it's just kind of an information analysis thing. And then you can, eventually, then you plug in the gene of interest, the microRNA, and then you get all these associated pathways because it's all by statistics.
So it tells you the likely outcome if you upregulate this gene,
It's
the probability of other things happening.
Yeah, it's like
if they're
throwing
talking to each
a
other.
rock into a pond and you get the ripple effect and all the different things. It's all, but it's not. It's all based on what the current literature is. So if something changes,
Right, it's probably as good as the computer program that's been put together. And they've got mathematicians. They're really not biologists, but they spend all their time thinking about this part of it. But it really, because we always, in the past, we talked about one thing and then you knock it out. And this is probably what the real world is closer to. And these things are fascinating. And you can just go online and download some of these computer programs. It's like crazy, but really cool. And just feel free to jump in at any point, because I obviously can barely get through this.
The key point of what you've said up to this point for allergy is if you change microRNA 21, it may be very important in changing Th1, Th2 balance. It may be a pivotal microRNA in that step.
At least in a mouse model.
Oh.
Exactly.
Are you going through the eosinophilic esophagitis?
Yeah.
Oh, okay.
Yeah.
It's been shown here.
Yeah, because that's what's been shown there in humans, and that's so we'll walk you through that. So these are called top cluster analysis, and it's kind of what Bill was talking about. It's kind of the next, where my little thing is, but this is blocked down here. But these are the yellow hexagonal things are the genes that are associated with interferon, and then these are the other genes. And then the red pathways are the ones that are connected to the interferon. And then these are actually the blocks or the mouse phenotypes that you might see. And so it's the same sort of thing about trying to really think on that kind of three-dimensional level what really is happening. There are lots of other microRNAs in experimental asthma. So as I said, the microRNA 21 stands out, but I added some others there. MicroRNA 126, upregulated in an animal model of acute house dust mite. LET7 is probably an important one. It's an IL-13 is a direct target of LET7, so it blocks that. Th1 cells have higher LET7 expression than Th2 cells. So that's probably important. And they actually developed these things, they call them AntagomiRs, which are blockers of the microRNAs, and you can see inhibition of LET7 by one of these significantly upregulated IL-13 messenger RNA expression of T cells, at least in vitro. MicroRNA 145 is upregulated in house dust mite models, and then another one, antagonism decreased experimental asthma severity. So there's a lot, in animal models, there's a lot going on in terms of, for asthma. In human subjects, as you can imagine, this is much more difficult. In an animal, you can give them the ovalbumin and then do it, and it may have to do when they're exposed to the timing of when they're exposed to an allergen or something like that. Where in human models, it's a little bit harder to do. And they took microRNAs from 8 patients who had mild asthma that wasn't in flare. They found no difference in expression, like 127 microRNAs. Another study where they took 6 pediatric patients that were having flare. They were admitted to the hospital, and they found a couple that were upregulated, but as you notice, not microRNA 21. And then another example, 16 patients with asthma versus controls. There were some microRNAs that were differentially expressed. But again, it probably, in the animal model, it's really nice because you just give them the allergen and you have this acute thing that you measure. So these and what they call AntagomiRs are a tool that they use in evaluating microRNAs, and they're really pretty cool. They're a class of chemically engineered nucleotides that silence the messenger RNA expression in vitro and vivo or both. An example is intranasal anti-microRNA R21 that they used in some of the mouse models. They'd spray it intranasally to see if they can block. It actually wasn't effective there. And they also don't always correlate with gene-deficient murine models. And it probably depends on what the antagonist looks like, how much of it really blocks, how it blocks the mechanism. But there are a lot of people that are very intrigued with these because this would be a potential treatment model for some of these if we really drill down on it. But in the real world, as Bill said, microRNA is actually upregulated in lots of different Th2-associated diseases. We found, and the one that's really exciting is eosinophilic esophagitis, but also it's elevated in atopic dermatitis, ulcerative colitis, and if you take human airway epithelial cells and treat them with IL-13, it will be upregulated. So I want to talk about eosinophilic esophagitis just a little bit. It's a great opportunity for research because the tissue's already available. You're making the diagnosis. You're taking the tissue anyway for biopsy. In 2 independent studies, microRNA 21 was actually found to be the most upregulated microRNA. And in one study, the microRNA levels inversely correlated with the esophageal IL-12 P35 levels. And when they've looked at these kind of bioinformatic pathway analysis, there's a correlation between T cell polarization and either Th1 or Th2. And that's one of the reasons why people are so and so here's another one of those heat maps. And here's the study they looked at normal people and then people that had active suffering with eosinophilic esophagitis. And what they found was that there were 21 genes here that were upregulated. And again, down here you can see that the red is upregulated and the blue is downregulated. And they had 11 genes that were downregulated. And right over there is 21. And then this is really cool because then they took people with chronic esophagitis who ended up not having eosinophilic esophagitis. They took the people with eosinophilic esophagitis, and then they took the people with eosinophilic esophagitis that treated with fluticasone and had a good response, and then they looked at them. And you can see that the eosinophilic esophagitis, you've got those 21 upregulated ones, and after treatment with the fluticasone, they actually significantly looked a lot more like the chronic esophagitis folks. And so that was really exciting for people that you could actually return the phenotype more to normal.
So, I mean, do they think that suggests it's a marker, or that there's actually some role of steroid inducing it and that being functional? It would seem the more obvious is that it's just a marker.
Yeah, I mean, obviously the thing that they were hoping is that you could use some of these as markers. So that you could actually, if you had it as a biomarker and you could say, elevated microRNA 21, this person has eosinophilic esophagitis, it would save a lot of time.
Did they ever go further and look at the different microRNAs and see if they're similar? Because you have 21, 146, 592. Do they look to see, are they similar targets or are they similar?
I don't know. I'm sure that there are people that are looking at it because they look really specifically at where it binds and what's conserved, what isn't conserved. So I'm sure that work's being done.
Is it being measured off tissue or blood or what?
Yeah. Tissue.
Okay.
A lot of those are targeting IL-13 genes.
Yeah.
And also some mucus genes.
Mucus genes.
So in the EE patients, you can't detect any of this just serologically. It has to be.
You can.
Oh, you can.
Yeah.
It's not just the tissue.
No, this was done in tissues, but yes, you can measure these in the serum.
What about the downregulated MIRS, like the 375 and 211?
Okay.
What about the downregulated MIRS? Can you correct the disease by giving them, or?
I don't know that they've done that, but they're clearly looking at the downregulate. Again, it's probably this IL. It's probably and when you look at those cluster maps that they do, the one goes up, one goes down, and again, that's why people are thinking it's that Th1, Th2 polarization. It takes more than one thing to do it.
They have a group of fluticasone non-responders and show that they didn't change.
I didn't see that.
They've got that when they've done that for microarrays for messenger RNA, they've shown steroid-resistant group.
Can you just give them the microRNA and it causes the disease? Is it causative or is it protective? Is it reactive or is it, what's its mechanism?
Well, it turns genes off.
Yeah, but I mean, is it cause the disease or is it protecting from the disease?
Yeah. Well, chicken and egg.
In other words, can't you just, if it's causative, you give microRNA to the animals and see if they get the disease? I'm sure it's not that simple.
Right, they're actually doing that sort of thing, and they've done it in early models.
You can induce the disease by giving it, so it seems like it's causative, or at least contributing to it.
It turns on a gene.
Yeah, but so it's contributing to the disease, not protecting. It's not reactive and protective.
Well, some of them are protective.
But this particular one, though?
This one might be. Yeah.
Is that what you're looking at?
But like in fibrosis models, they're thought to be pathogenic, like IPF.
Well, it's just like the eosinophils. Remember, way back, you didn't know whether they were protective or they were pathologic, and I just wondered.
Then they looked at, I think most literatures in cancer, they associated a bunch of lymphomas and leukemias with overexpression of some of these microRNAs.
But you do assume that it can be protective in cancer, too.
They're looking at both, because different cancers have overexpressed microRNAs and underexpressed, so could you give underexpressed and change the course of the disease? Maybe.
This is just a map of the microRNA.
Right.
If you get a map of the person's DNA, it has to be the interplay between the two. I presume this is regulating gene expression. And if you do a similar map of people with eosinophilic esophagitis or chronic esophagitis, you see a different heat map of action.
Yeah. So this is the heat map for chronic esophagitis, and you see it as a different heat map.
Kind of related to Gary's point, have they taken like esophageal tissue and tortured it with IL-5 or something? I mean, is this a downstream issue or is it somehow a primary problem? Is what I'm wondering.
Yeah.
And in other words, can you create these changes in the microRNAs with something upstream?
Yeah, and I don't know. I didn't read a paper that looked at that, but I suspect that they're looking at all kinds of tweaks. Oops.
Well, they've done that before and after treatment with steroids. I mean, these are all primary biopsies.
But again, that regulates IL-5. Doesn't answer this question whether is this a downstream response to something else torturing the cells? You know what I mean?
Working in the field showed upregulation of taxin genes. So I would assume these things, treatment might, among other things, downregulate the expression of making eotaxin.
And again, when you look
at
at
mechanism on that primary abnormality.
So when you look at like the interferon gamma and the IL-12, these things, they work together and have all of these just diffuse other connections and responses. And I don't know why these slides didn't turn out, but these were a couple that showed there were different microRNAs that were upregulated in eosinophilic esophagitis, and I actually don't have the slides, so I don't know what those were. I don't know if they showed up in your slide.
The first one is 21.
Yeah.
Next one is
Look at it.
70.
No, it's okay. You guys can look at it. All this really shows you is that there are different ones that are upregulated, and then other microRNAs that are clearly downregulated in eosinophilic esophagitis. It's weird. Some of these just did, and others did. This one, again, shows a decrease almost to normal with a couple of these when you had eosinophilic esophagitis that was in remission. And so obviously people are really interested in trying to find microRNAs that last long enough in the serum that they could be good biomarkers. And they took a couple of these that were upregulated. And you can see that the microRNA 146 in an A and B positive and negative predictive value was not all that bad in differentiating normal versus active eosinophilic esophagitis. but it wasn't quite as effective when you looked at in remission, separating the eosinophilic esophagitis from remission, sort of like flipping a coin. But there are labs that are looking at this with that whole vast array of microRNAs, looking for things that might be potential biomarkers where we could actually make a diagnosis. And it is an exciting idea. Anything to add to that, Bill?
I'm sorry.
Anything to add to that?
Just that's probably the big areas. Since they're stable in the circulation, you can easily draw them. And even though the assays are currently complicated, once you validated this as a biomarker, then you could do RT-PCR, which would be very cheap to get the same info. So you could use this as a screening tool to see who would be responsive to the therapy, or so that's the excitement of it, because that's where it's been borne out in the cancer literature. They've got panels of these now that can make pretty accurate predictions of who's going to respond to a particular cocktail of cytotoxic agents, for leukemia.
I mean, is there a good reason biochemically to believe these will be good biomarkers? There are lots of things in the serum that are, stable, but do they actually know, are these having distant endocrine-like effects throughout the body, and they're therefore going to be easy to measure, or are these just sort of,
I think if you look at something like microRNA 21, if it really is one of the important things in the switch, the polarization from TH1 to TH2, that makes it more than just something that is floating in the bloodstream, right? It makes it really pretty exciting.
And this will also get more refined, because like you said, those initial mouse was like 500 microRNAs. There's now like 2,000 identified.
Yeah.
There's like 2,500 human ones, but that's gone up maybe 50% over the last 3 years. So, I mean, it could be like, at least it's a manageable number.
Yeah. For you. Have they looked at like eosinophilic leukemia or chronic hypereosinophilic syndrome and look and see if these same markers are present?
I'm sure people are doing that. There's a huge cancer literature. I read a couple of review articles, but there are people, just labs that are just, tons of labs that are working on this stuff.
Yeah, for every, I'd say, allergy article, there's 100 to 500 cancer ones.
Yeah.
Or 1,000.
He's already employed
It's amazing how many
cancer.
I think so,
To diagnose.
Yeah.
It's going to respond to what drug?
Yeah. So you can imagine, because some of these cancers now really, it's very personal and it's on a gene level. So stuff like this is, really exciting in terms of, and if it actually would help you understand the response to treatment, it's really exciting. And this again just sort of shows the same sort of thing. Here's two there's microRNA 223 and microRNA 21, which are upregulated in eosinophilic esophagitis, using the same sorts of mathematical models to kind of do some predictive. They're co-regulated. What would that look like? What are they turning on? What do you think about? And then this is the next iteration of that again. So when you kind of trying to put it all together, up in the right-hand panel, you've got the microRNA 21, and then you have below that the IL-12 P35, and that isn't really, it's got messed up again. I don't know how this happened, but over on the right-hand side, if you had it on the paper, there's that dark area, which is the highly conserved nucleotides, and that's where the MR21 binds to the IL-12. And if you think about it in this big picture, you've got your allergen. Looks like that got blocked too. Okay. Well, probably your pictures are better, but so the allergen tweaks the system. You get upregulation of microRNA 21. You get the switch over to the which you can't see on. Look at your pictures. You get to switch over to the TH2 profile, and you can see over there the eotaxins and eosinophils as well. But that's sort of putting it all together. Just quickly, since we're running out of time, atopic dermatitis actually has kind of a mixed TH1, TH2 inflammatory response. Like the other inflammatory diseases we've talked about, microRNA 21 is elevated, but also 146, 223. And the most upregulated skin is actually microRNA 155, which actually suppresses cytotoxic T lymphocyte-associated protein, and it kind of does this balance with Th1, Th2, so it actually makes sense. And it also targets IL-13 receptor. Allergic rhinitis, there have been some small studies done on that, and there the take-home messages are pretty confusing. The Chen article that was done in 2010, they found downregulation of microRNA in the cord blood samples with increased TGF-beta receptor in the cord blood, people with these increased IgE. And they did another study where they took the kids that were like 6 years old and had allergic disease, and they had downregulation of microRNA 21. So it's the flip of what we've seen in the animal models. And the question is, does it have to do with when you got the allergen challenge or where you're at in the process? But allergic rhinitis is the one that doesn't really fit the rest of the pattern. And this just shows you a core set of microRNAs that are involved in allergic inflammation. And you can see the targets there, the LET group, all IL-13, and then microRNAs, the target is IL-12. So again, making you think about that polarization. They've looked at eosinophils, mast cells, and T cells, and I didn't go through that literature. There's a fair amount of literature on that, but they clearly play a role in the differentiation and proliferation of allergic cells. And then this last picture really, they took a lot of the other microRNAs and where they block, and it kind of puts the whole thing together as you start thinking about this Th1, Th2 polarization. And this was just published probably at the end of 2013, and is probably completely out of date. There's probably a much more complex diagram if we were to take a look at it today. So just in terms of future directions, I think understanding more about the ability of these guys to coordinately target common pathways, and that mathematical modeling, I think, is going to be really important in the future. And looking at things like this polarized T cell response, what turns it on, how can you go there, how can you go back from that? The possibility of using these to target genes and reverse disease phenotypes, and as Bill said, a huge amount of work being done on that in cancer. The interplay of microRNAs and epigenetics, because that would be a really fast way that you could change things. Therapeutic potential of the mimics and the inhibitors, which they're actively looking at as well. And then probably the thing that will be closest to being marketed is the use as biomarkers for diagnosis and response to therapy. So even though it's probably not quite ready for prime time in allergic disease, it's getting close, and people are just cranking out data all the time.
Yeah. The way you present this, it strikes me as important as knowing your DNA. I mean, this is so critical to regulating your DNA. Where was this all these years?
Well, that's a good question. I mean, do we think that Lucy had these mechanisms?
Well, that's the other question. How far back in evolution? They found it in the roundworm, but I mean, does every organism that has DNA have microRNA?
Yeah, they're in plants. They're in single cell yeasts, so they go back pretty far evolutionary. The peewee ones aren't as conserved and probably aren't well, they're important, but probably later evolutionarily, I think.
Where'd the name peewee come from?
I don't know.
Baseball. It might not be in our world, but probably in Bill's world. I mean, do they have knockout models for these now? And see what happens if you knockout?
Yeah, they have. I
that
showed you
lethal?
that in that one thing. That was.
Is that lethal, or can they not get allergic disease, or I mean, what's the story with the knockout?
It depends. They make these mice. So in that particular one, that was a knockout for 21. In some of the knockout models, it's fatal to the embryo. There are knockout models, as I described, one of them where the animal developed fatal autoimmune diseases. So these knockouts can have major effects.
So there's some good function to this as well as bad function.
Yeah, there would be like key for development. Certain organs would need it. So if they could get away, they're working on that because you can get conditional knockout, so they have it when they're young, and then when they're an adult mouse, then you can have it knocked out. At that point, then you can see what effects it might have on.
Are these big enough you can target a protein at them, or are they too small for monoclonal?
small like that.
So you couldn't use a monoclonal antibody against these?
No, but you could. But these other things are better because they're smaller. They're easily delivered. So that's the excitement there for the drug industry.
Yeah.
Because these enantiomers are tiny and they're easily delivered by a nasal spray, or you could have a patch or whatever. So you wouldn't need a big monoclonal antibody.
Yeah, they're chemically just making these little blockers.
Down to
And the
nanotechnology.
key thing is different blockers will probably conformationally result in a differential kind of a block. So some of them don't correlate with the knockout mice, and it's probably because you just engineered the wrong little block of microRNA.
I thought a lot of this at the beginning was for diagnosis. They had these little arrays put on chips and stuff for this microRNA. Is that all gone now? That they don't do that anymore for diagnosis? They just have these things floating around now?
No, I'm sure they'll do that with, I don't know
500
how
or
they'll do it
600
technically.
little markers on a chip, and they put some cells in there to see what they have.
The problem with this whole thing is the information that's generated is incredible. So you need a lot of bioinformatics, computer time to figure it all out.
The primary care physicians are going to be doing that and diagnosing and treating these diseases.
You wouldn't go that route. You would, once you figured it out, then you'd do like an RT-PCR, which would be very cheap. If you targeted it to maybe just half a dozen things, then that could be easily. This would be way too expensive. I mean,
You can see that research these days is not for amateurs.
Or for,
And that every good lab almost
or for
needs
poor people.
giant computers and mathematicians to do this sort of work.
So, a technical question. When you scan documents into a PowerPoint presentation, you scan them, it'll try to interpret it as text unless you scan it as a JPG.
That's
So
what I
you
did.
make it a JPG, you get a picture of what you actually scanned.
That's why I cut.
Yeah. This happens a couple of times.
So you did a brilliant job.
So for those of us over 60, this is not going to make it to the end of pretty much, right? But for Matt and David, this is important. But for the rest of us, it's totally unimportant. Is there more Star Wars blowing through us?
I was thinking about it this morning. You and I were rarely in the pod, so we'll talk about it.
If one of you is there, just like you're saying, do resources and so that they can figure it out. I mean, obviously you guys here on Tuesday. That's got to be my guess.
There's light at the end of the tunnel.
Let me follow up with that 48 hours.
You still think Monday's be a good day though?
I think Mondays are potentially the best just because we're both he's here. So that's a problem.
So that's a commercial thing you've got purchased?
Yeah.
That's actually but yeah, it's definitely still on my. I have a flag on my email.
Well, we have some stuff going on, and I want to get you to sign that medical information. And I can get you set up with one of ours myself.
It's always
So you can set up some research and all that stuff. So that's I can't do that. That's an MSL.
okay. Well, let me see what Drew is.
Just going to go to those
Yeah. All right. Like I said, we have to just coordinate.
Oh, no, it's
So I'll see you out there.
so the 19th I'll actually do it. Plus the next one.
Yeah, you can come down sooner. I don't.
So we're looking at
very
Well,
much
but we
time
could,
you have.